Edgar Vega. I'm the Associate Director of Patient Advocacy for a company called uniQure. And so we're going to be focusing tonight a little bit about gene therapy and Huntington's disease. No? So I'll start with that. But to start, I'm going to focus a little bit on the history of our company so you get an opportunity to get to know us. And so in 1998, our company was actually established as a company called Amsterdam Molecular Therapeutics. And so this company actually started in the Netherlands, which is, so we were the first company in Europe to actually launch a gene therapy in 2012. And so en 2020, en 2020, nosotros empezamos el proceso para un ensayo clínico para condiciones de Huntington's disease en el 2020. So in 2020, we actually dosed our first patient in 2020. Ahorita yo creo que las personas que están haciendo la traducción donde está diciendo que está haciendo esto. Bueno. Ok, so en el 2020 empezamos a dar la primera dosis para nuestro ensayo clínico de Huntington's disease y en el 2022 nos aprobaron el primer medicamento para una condición que se llama hemophilia B, que también es genética. So in 2022, we actually got our first product approved for gene therapy for hemophilia B, which is also a genetic condition. We have 3 sites throughout the world in Amsterdam, which is the area where the majority of our research is done. There's about 290 employees. That number tends to change very quickly. And then in this area just outside of Boston, called Lexington, Massachusetts, we have approximately around another 300 employees. And that's where actually all of our manufacturing is done for our products, right? So our clinical trial products, and then now the hemophilia B product that just got approved as well. And then our CEO is based out of Boston. I work out of the Boston office, but I actually live in a different part of the country, and I'll explain that in the next slide. And then we actually have a very small office in Switzerland, in Basel, that is 10 employees, and that was actually a company that we acquired in 2021. In total, it's about 500 people, with the majority of those focused on manufacturing, quality, like those things. We are a gene therapy focused company. So nosotros somos una compañía que está enfocada en la terapia clínica. Es lo único que hacemos, no tenemos otros productos. La ciencia que nosotros proveemos es específicamente para eso. So we just focus specifically on, and our lead product is right now this Huntington's disease, but we also are focusing on other neurological conditions such as ALS. I don't know if the Parkinson's one's out there anymore, but temporal lobe epilepsy, Fabry, so different genetic diseases. And so one thing that I like to educate people about is patient advocacy, right? So what role we play within the pharmaceutical company or biotech company. And so there are 2 people that actually work in patient advocacy at uniQure. So one of them is my manager, Dan Leonard. So you can see him on the right there behind the orange. And then I'm on the left. And so my name is Edgar Vega, and I actually live in San Diego, California. So I'm super excited to be here with you all. And it's my first time in Puerto Rico, so I'm super excited. And it's been fun. So it rained a lot yesterday, I will say. So that was different. Yeah, pretty much that's who we are. And so we love working with you all. Being part of these events is really what I like to just be at, right? Like talking to you all, getting to know you yesterday. We had a great day kind of getting to know the foundation and everything that you all do. And so then we actually try to make sure that our programs are in line with what the needs of the community are, right? And we do that through different meetings. There's a picture of Seth, actually, that's sitting right there. He was a patient speaker about 2 years ago, I think. Yeah, about 2 years ago, right? Like out of our Lexington office. That's really what we do. Next week, we're actually having, for HD Awareness Month, we're having a patient speaker in our Amsterdam office come and talk to our employees, right? Because we want our employees to know exactly that what they're working on has an impact, and they know that, but really getting to know people from the community is really where we try to keep that engagement level really high. But we're all inclined for that one mission, right? And so that mission is really to deliver one-time transformative therapies for patients' lives. So that's our mission, that's our driving force. And we hope to really be able to accomplish that through our clinical trials and hopefully get different products for genetic condition. All right, okay, I guess the top got cut off, but we're going to delve a little bit into the science. I'm not going to focus so much like on the ins and outs of our clinical trial. This is going to be a little bit more science focused and like what it really takes to hopefully bring a gene therapy product to market, right? So if you have questions about like our clinical trial or anything like that, please come and see me. I'm not that hard to miss because I have an orange shirt on, so please. But let's start a little bit just basic science. Every cell in our body has 46 chromosomes, right? And so you'll actually see that in the little black picture right here with the colors, right? And so we all have 46 chromosomes, and each one of those chromosomes tends to code for a very specific thing and for a function in our body, right? And so that actually helps determine the color of our skin, how tall we are. So some people are short, some people are taller, the color of our eyes. And there's even thoughts that it could actually help influence like things like intelligence. But it also sometimes can carry information that can cause different genetic conditions, right? So I had mentioned hemophilia, that's a sex-linked X-linked chromosome genetic condition. HD is another one. Race, all those sometimes can potentially cause a specific genetic disease, but they all have codes, right? And so the reason that those codes kind of change is what we call I was going to say in Spanish, mutation o una mutación, ¿no? Entonces, esas condiciones genéticas a veces saben por una mutación que se encuentra en el código de las cromosomas que nosotros tenemos en nuestras células. Yeah, so this genetic component is linked to health and disease, right? We know that some of these conditions end up coming out in that code. So I'm trying to not look back. All right, so then what do? So we know that the genetic conditions exist. There's a way that we can utilize science in order to try to help manage some of the symptoms of this disease, hopefully manage the symptoms of the disease. And so gene therapy is a technique that uses genetic material to treat or prevent a disease, right? So we know that there's this thing that happens in the body, and we can utilize science to hopefully not have that thing occur, right? So how do we do that? So we can actually take little small pieces of components of DNA and help engineer them with things that we find in science, right? There are different, what we call vectors, or they can be viruses, plasmids, a whole bunch of different things. And then hopefully deliver those to different tissues of the body that can hopefully help treat the genetic condition, right? And so then your body naturally starts to make the production of that molecule, or what we're calling a therapeutic molecule, okay? So it's going to sound a little bit of a different way. Oops, did I go back? All right, so this code that we can engineer is essentially a recipe, right? So it's a recipe for something that we want to be done in the body. And then the next step is we have to figure out exactly how to get that recipe inside the container, okay? So think of it as like a shipping box. If you order something from the store, it comes in a box and there's something inside, right, that you kind of want. So think of it as the same thing. And the way that got there usually was like a shipping truck or on a boat or an airplane, those types of things. So in order to try to make this a little bit more manageable, think of like gene therapy as that potential. So there's something that's inside a box. The box is important, and then the way that it actually gets to you is also very important. So those are the three things I think that I would like you to think about as we talk through the next couple of slides. How do we do that? So the first one is there's like a whole bunch of different ways of gene therapy and kind of what gets accomplished. In some diseases, we want things to be moved down. In some diseases, we actually want things to be moved up. And then there's some diseases where we actually just want to take out parts of that code that cause that disease. And then how do we do that? So like I had mentioned, there's a whole bunch of different like shipping containers that we could potentially use to make that accomplished, right? And most of these tend to be pretty high science, but I'm just giving you a little bit of a general overview of how we kind of do that. And then the next question is, how do we actually do that? Do we do that inside the body, or then do we do that outside the body, right? And so there are different ways that kind of like work on that they could potentially utilize further. And so our company actually focuses on making the body create something, or actually making the body not create something. Okay, so that's what we as a company are focused on. And we actually focus on a very specific type of gene therapy, which is called adeno-associated viral gene therapy or adeno-associated vector. And then we actually try to do that directly to the patient, right? So we're not utilizing some sort of genetic material to do something outside of the body and put that back inside. What we're actually doing is doing it directly in the body for a patient. So the concept of AAV gene therapy is not new. In about 1965, they discovered these adeno-associated viruses in 1965. And so then in 1993, that first AAV-based gene transfer, so that little recipe that I was talking to you about occurred, right? And then there was a trial that was firstly done as a potential treatment for genetic diseases in 1996. And then they actually discovered that there was a whole bunch of different boxes they could utilize for this specific AAV. And then that each of these boxes can actually help different organs in the body potentially, right? So there are some like the brain cells a little bit more, some like liver cells a little bit more. And so then that kind of process started a little bit to be discovered more. And then that actually led to that product that I had mentioned back in 2012, which was the first AAV-based product to be approved. And then in 2017 in the US, that was the first, there was another product that was approved, but this time here in the US, right? And so now there are actually 5 AAV-based gene therapies that have been approved by the FDA, and there's 6 here. From 1965 to now, we're starting to get to an area that this could potentially be, that this is actually a reality, right? That this science is advancing, and it's kind of cool. I'm not gonna lie. So this is just a little bit of an infographic to give you an idea of like the different indications that this is being used for. So there's hematological conditions, there's central nervous system diseases that this is being investigated, oncology. And again, this AAV gene therapy is just one of different ways to think about the science. Okay. So what really goes into the development of a gene therapy product, right? So again, we're going back to that recipe, right? So we want to actually identify a gene that we could potentially work on, right? And then we have to figure out a way to get that shipping box container that I was talking to you about. And then from our perspective, from company perspective, is we want to actually make sure that we can manufacture the science that we're doing, right? We have to figure out a way to do that. And then once that's done, we can also try to figure out how we're going to do this. Is it going to be outside the body? Is it going to be inside the body? Right? Cool to think about. So the first thing is this gene. Okay. And we know in Huntington's disease what causes Huntington's disease, right? We've identified that gene a couple of years ago. We know that we could potentially do that. And so again, this like the CAG repeats are important. And that's essentially what we're trying to do. Our philosophy is interesting because we're actually going after the 2 parts of the brain that tend to be affected most, which is the striatum. And I just wrote out the other one because I got my life a little nervous. Right, the caudate and the striatum. So these which are these deep structures of the brain. So that's going to come into play a little bit later on when we talk about the actual delivery of this, right? So we know the gene. It's something that we could potentially work on. So then like the hypothesis starts, right? Could we utilize this type of science for this specific genetic disease? And so then that science starts to take shape a little bit, right? So what behind me in that picture is actually these adeno-associated viral vectors that I'm talking to you about, actually go into glial cells and target neurons. And so you start seeing them go into like different structures of this model brain that we have, right? So we're like, okay, like this is the next step. This looks like it could potentially work. And mind you, I'm like giving you like a 15-minute presentation, but this takes years, right? So this is like a very slow process. But these are pretty awesome kind of scientific breakthroughs. And so then as the research is advancing, we actually have to continue to what we call upscale. So make more of a specific product in order to move forward in like the science process. So it really starts with what's called a small bioreactor. So it's like a 2-liter size of Coca-Cola, right? So we start something that small, and then eventually we get to a very high like that bioreactor that holds anywhere from 1,000 to 10,000 liters, but that's actually once the product gets approved and we start making a lot of products. So right now with our clinical trial, we're sitting between that 50 and that 500 liter range for production. Again, this is just to give you an idea of kind of what happens behind the scenes as things are moving forward, but we want to make sure that the product is repeatable, it's safe, it's effective, right? And so there's one of the reasons that there's a lot of manufacturing people is for this exact reason, right? And so then we have to then also at the same time that this is happening is deliver this. How are we going to do this, right? And what shipping container? Is it going to be a boat? Is it going to be a truck? Like, how are we going to actually get this to where it needs to go? So hopefully the animation plays. But you can actually see here, it's another visual that the AAV5 or that medication that I'm talking to you about actually makes it to areas of the brain where we want, right? So as you can see, they're like in deeper structures in the brain. And so how are we going to accomplish that, right? If you want specific details, I can talk to you a little bit about that. But you can see through this model that the drug is getting where it's supposed to go, where it's supposed to go and where we intended. So that's kind of summarized a little bit by this. I think if you've worked in neurology or any central nervous disease, you know that this is very complicated science. This is difficult, right? There are so many things that go into this. As you've seen by some of the presentations, it's just a lot of different pieces to the puzzle, right? But we want to just make sure that we're getting where we're supposed to, how are we going to get there, and then that it actually works, right? And it does what it's supposed to. But it's a pretty challenging area of that. So how do we get there? So one of the things that you have to know about science in general is that models become very important, right? In that. So we actually start with very small, we call them petri dishes usually. And so then we'll actually grow cells from that. And then as trials progress, we want to make sure that they're safe and effective in that specific thing. So how does that happen? Like mouse models are super important. We've done studies on guinea pigs, right? So smaller, more human-like, quote unquote, animals, and then also like human primates. And then once those things are safe and effective, then we actually move on to humans, right? And so we're not just going to go ahead and do that just out of nowhere. We actually have to go to the government to go ahead and give us the okay to go ahead and do that. But we actually just want to be able to help reduce time, right, for development. And then also just reduce the number of animals that are needed. And obviously study the safety and efficacy aspects of human cells, right? Because that's the most important thing, because that's ultimately where we want to go. So how do we do that? So we actually start with skin cells, and this is from an HD positive patient, right? And then from there, we'll actually grow them and amplify them, and then they'll actually become brain cells, or they'll actually become little small brains, okay? Which is cool if you think about it, right? So we can actually culture these things and grow these things. And then the green picture that you see back there is actually that AAV5 or that medication actually go into those different small structures of the brain. And so then we're like, okay, hey, like we're actually seeing that this works. And then from there, that process that I was talking to you about before tends to go on and on. So again, cultured human neurons, that picture that I was showing to you before, we're always testing for safety and efficacy. And then that kind of moves on to the next like mouse model, right? Which is what we're talking about. And so again, safety and efficacy. And then we move that on to larger animals that tend to be a little bit more human-like. And again, safety, efficacy, and then now we can actually start to see where is the drug going, right? What is happening as the science is continuing? And so then if that shows promise and it shows some sort of capability to hopefully be effective, then at that point we actually go to the government and we try to apply for a clinical trial, right? And so all the data that we had seen previously, and again, I'm talking to you about years of research. We'll go to the government and we'll say, hey, we want to file what's called an IND, so it's an investigational new drug application, and then actually start clinical trials based off of it. And so in ours, we started in 2020. We're about 3 years into it. So hopefully next month we'll have enough as to what some of the data looks like in humans. And we're super excited about that. And we'll definitely keep you up to date as to what's going on. But really, the firm idea, right? So, okay, we have this science that we think can help this genetic condition to hopefully get to approval. It takes a really long time. We're talking anywhere from 10 or 12 years. And there's a whole team of people that kind of work on this day in and day out. People that have made this their life's work, right? And that's one of the things that I think I would like you all to walk away with is that a lot of us are really passionate about what we do and talking science and getting to know people. I'm in a very privileged position to get to share that with you and come to events. And I think these people are the people that work on our hemophilia B approval. But honestly, from the bottom of my heart, thank you so much.